Good afternoon, everyone. Um, I would like to start by welcoming our audience connecting from all over the world, students, CAUS community, in Kingdom universities, organization and attendees, and live stream audience on CAUS social media. Our session today is on advancing science and technology innovation by crossing the art science design interface. And today we have the pleasure of listening to Professor Donald Ingber, a leading scientist who has so beautifully made a breakthrough in advancing human health care by breaking down the boundaries between science, art, and design. This manifested itself in his pioneering work to mimic the mechanical forces and shapes of human organs on a chip. This human organ on a chip technology was named Design of the Year in 2015, honored as one of the top 10 emerging technologies of 2016 by the World Economic Forum. Most importantly, the artistic beauty of the design granted it a position as a permanent collection in the Museum of Modern Art in New York. On the career side, quickly, Professor Ingber is a professor of surgery and professor of bioengineering at Harvard, and is the founding director of the Wyss Institute for Biologically Inspired Engineering at Harvard. He is member of the National Academy of Medicine, National Academy of Inventors, American Institute for Medical and Biological Engineering, and the American Academy of Arts and Sciences. In the spirit of the focus of this session, today we have a special guest, Joe Watkins. Joe is a live artist from the Sketch Effect. He's joining our conversation today. He is from Ohio in the US. He will be actively listening throughout today's lecture and bringing what is shared to life in a visibly and visually compelling and memorable way. Without further ado, please join me in welcoming Professor Ingber. Thank you.
I'm going to share my screen here. And can you see that? Can you see my screen? Fantastic.
Yes.
Thank you so much. So as you heard today, I was asked to speak about advancing science and technology innovation by crossing the art science design interface. As you heard in the introduction, I'm a professor of surgery at Harvard Medical School and at Boston Children's Hospital. But I was never trained in surgery. I'm a professor of bioengineering at Harvard School of Engineering and Applied Sciences and the founding director of the Wyss Institute for Biologically Inspired Engineering, but I've never had an engineering class in my life. So the question is, how did I get here? And I've got here by taking some very interesting paths, and I think that's what I'd like to share with you today. So it all started actually in an art class. I was a Yale undergraduate. I was a science major, molecular biophysics and biochemistry. And usually these are totally separate worlds, at least in America, and almost separate campuses. But I happened to see students walking around holding paper structures that they constructed that looked like the bottom right, which to me looked very much like the viruses that I was studying in my classes. And I asked these students, what class are you building these? And they said it's called three-dimensional design. Now, this caught my interest because at the molecular level, I was learning everything was three-dimensional shape dictating function. And so I actually got into that class, and that is where I was first introduced to a building system, a form of architecture that's known as tensegrity. These are structures who get their shape stability through continuous tension or tensional integrity, tensegrity, rather than through continuous compression. So think of Stonehenge. You have multiple stones that are stood together, other ones lying on top, held together by gravity, and they have form. But if they were to go into outer space where there's no gravity, they would just float free. In contrast, these structures self-stabilize from within, And they were first visualized by a sculptor by the name of Kenneth Snelson. And these are 16-foot long steel girders that do not touch, but are pulled up and open, as you can see at the right by my little sculpture, by a continuous series of tensile strings that connect it and pull it open. And Buckminster Fuller was the first person to really describe this system before he could even visualize it. And this underlies why his geodesic domes were so incredibly strong and efficient. Artists have also recognized that this is the way our bodies are built, because if you see a skeleton in the anatomy lab, you'll see 206 bones that have to be wired together and hung from a stick to look like us. But in reality, we're pulled up against the force of gravity because all those bones are interconnected by a continuous series of tensile muscles, tendons, ligaments, and fascia that, again, pull us up against gravity. And it is the tone in my muscles, or the tension in the system, that determines whether my arm's stiff or flexible, and that is tensegrity. Now, in this class, the teacher happened to make a model with elastic strings, much like the one I show here, which was round, and as he was talking, he would flatten it, and then he spread it and let it go, and it bounced up in the air exactly like that. Now, this caught my eye because the same week I was learning how to culture cells in an anatomy lab, in a cancer research lab. And cells, when they're on a dish, look as shown at the right. They have a central nucleus that's round, and the cell and the nucleus is flattened and like a pancake, and the cell is polygonal. And when you try to pass the cell from one dish to another, you clip the anchors, and these cells spontaneously pull up, round up, just like that model. So I immediately assumed that cells must be tensegrity structures. I went back to the cancer lab. We used a drug that calls cells to round up. I said, oh, the tensegrity must have changed. And the postdoc, and I was an undergrad at this time, I was 18 years old, and the postdoc said, what did you say? I said, tensegrity, Buckminster Fuller, sculpture class. He said, never say that again. So I shut up and I went to the libraries. And as they say, that's the beginning of the rest of my life. Now, this is the late 70s, mid to late 70s. At that time, people thought of cells as basically a gooey, viscous cytoplasm surrounded by elastic membrane, almost like a water balloon filled with molasses. However, the first papers were coming out that identified that cells actually have an internal molecular skeleton known as the cytoskeleton. That's literally a framework of molecular ropes, cables, and struts on the nanometer scale that go from the nucleus to the surface. So this began to take shape in my mind. I built my own models. These are aluminum struts taken from a chemistry lab where you hold glassware, high tension cables from a sailing store. I made a nucleus out of sticks and strings, attached it to the surface by black elastic threads. And now you can see when the cell spreads, the nucleus spreads. And when it's round, they both round exactly like we see in living cells. I won't go through the whole story, but this culminated in my 1998 Scientific American paper, where I described what I called the architecture of life, that there's actually a universal set of building rules, which is tensegrity architecture, that guides the design of organic structures from the simplest carbon compounds like buckyballs to complex cells, tissues, actually organs and organisms. And the bottom you see in this paper, I first proposed that proteins are tensegrities because there's stiffened regions, the alpha helices, and then attractive hydrogen bonds, which are tension. And we've now, and others have confirmed this both computationally and experimentally. Now, also as an undergrad, at the same time, I was taking classes in developmental biology, where I'd see movies as similar to the one on the right, which is a zebrafish. This is the yolk sac, the egg just divided into 2 and 4 and 8 and 16 and 32, and the cells are getting smaller and smaller as they parse out cytoplasm. But then it hits a point where the cells generate tension inside their cytoskeleton, pull on each other, and you literally see them sculpt the different organs that form the embryo, the brain, the eye, the spinal cord, et cetera. So that mechanical forces are exerted throughout development, and that they may be as important as chemicals and genes, which is what a turn of last century, people only talked about mechanics, but that was sort of the baby was thrown out with the bathwater as people got more reductionist. But to me, I could see that maybe this may all have to link together. Now, if you take cells out of tissues, you trypsinize them, you put them on a dish, they'll spread, as I showed earlier, like at the right by immunofluorescence. But if you put them on a flexible substrate, that tension, I'm sorry, that cytoskeleton is contractile. Actomyosin generates tension and the cells will pull the substrate up into wrinkles. And if you relax the tension, it relaxes back. So you have to think of cells as almost like a bow and a bowstring. It's a dynamic equilibrium. It's a state of isometric tension. They're pulling against their adhesions to extracellular matrix, which is in the body, or matrix-coated substrate. And you could think of the cells attached to their matrix almost like eggs sitting in egg carting. It's their anchoring scaffolding. It orients them. It tells them what's top and bottom, polarizes them. Now, again, late 1970s, Judah Folkman at Harvard published a paper in Nature, a really fundamental breakthrough paper that suggested that the shape of the cell, whether it's round or flat, will determine whether it grows. Now, people thought it was phenomenology. They argued, well, you don't know, there could be different matrix molecules or different growth factors being released. But it suggested the idea to me that actually the shape changes that I was seeing through tensegrity in mechanics could control function. And finally, in the late 1970s, Mert Bernfield was looking at development, and what he found is that that anchoring scaffolding, the extracellular matrix, or ECM, shown here in green, that the way cells form patterns or branches and morphogenesis in different tissues is that a subset of the cells grow in red and bud out. But what he found is that the matrix is stiffer than the cells, so they have to actually remodel that scaffolding. And the first thing you see before growth up here is a thinning of the matrix due to degradation of the molecules that make it up. Where there's degradation, you see increased growth. And then the degradation is matched by even more synthesis. So you're getting net expansion of the scaffold, which is beautifully matched by net doubling of the cell number, which leads to budding. So with my vision of the mechanics in this, what I thought was that if you get thinning of the basement membrane, the extracellular matrix, and the whole structure is under tension to begin with, isometric tension, because all cells on both sides of it generate tension, then this would be like a weak spot in a woman's stocking, and it might stretch out more than the rest. And if the cells are adhered to it, they'll stretch out. And if Judah Folkman was right, then stretching would stimulate growth, and this would be beautifully coupled and give you morphogenesis. On the other hand, I was working on cancer, and if in cancer, you see the same thinning of the matrix before tumor formation, before cancer formation, early stages of tumor genesis. And the idea here is that that would also stimulate growth. But if you didn't keep up, if synthesis of new matrix was either the same or less than new deposition, you wouldn't get expansion of the boundary, and instead of budding, you get a pile, and that was what we call a tumor. And if this deregulation increased over time, you'd get degradation of the matrix and breakdown, and that's what we call invasive carcinoma. And this was the last chapter of my PhD thesis, and I, again, not time to go into it, but this is all shown to be true, and mechanics and cancer is a very hot area right now. So at this point, I moved to do a postdoc with Judah Folkman because he was the absolutely right person for me. He worked on cell shape, but he also worked on cancer. And he was the brilliant luminary who came up with the concept that tumors must stimulate new capillary blood vessel growth, or what is angiogenesis, in order to grow. That led to multiple drugs that now work for cancer as well, like Avastin, as well for things like blindness that also involve abnormal blood vessel growth. And within a year as a postdoc in his lab, I had another serendipitous experience, another aha moment, where I had a fungal contaminant in my dish of capillary endothelial cells. Now, normally all the cells just pop off the dish and die. Anyone who's done culture knows this. You put the dish into an autoclave to kill it so you don't spread contamination. But what I saw was this. There was a fungus beneath it, the cells popped off, but there was a gradient of cell rounding at higher magnification. So they were round beneath it, but still adherent, completely normal on the opposite side of the dish, and you got a gradient of cell rounding. And because I thought cell shape was important for growth, I cultured this fungus. Long story short, again, it led to the discovery of a molecule that was eventually called TNP470. It's the most broad spectrum angiogenesis inhibitor ever identified. It synergizes with chemotherapy and radiation therapy. 20 years ago, it went into human clinical trials. The first complete remission in a human with metastatic cancer was in New England Journal in 1998. But the trials were stopped partly due to neurotoxicity, partly due to financial issues with the company. But the point is that that ended. It was like a painful failure. It didn't enter phase 2. The good news is that another small company took this compound, put a polymer on it so it won't cross the blood-brain barrier, and that has just completed phase 1, looks very exciting, and they're about to start phase 2. So it's never over till it's over. And this is where I got introduced first to the world of patents and clinical trials. Now, I was in Folkman's lab as a postdoc, as were Bob Langer, the famous bioengineer, and Jay Vicanti, who was a transplant surgeon doing research. And they recruited me to, as they were getting into a field that they were beginning to pioneer known as tissue engineering. And we helped to initiate the first studies with biodegradable polymer scaffolds. I was introduced to chemical engineering and material science for the first time, much more experience with patents. And we together started one of the first tissue engineering companies called Neomorphics. And so this was how I got into the world of biotechnology, consulting, and a whole other type of the world of a scientist today. Now, the tensegrity model was always in my mind, and what it predicted to me is that if forces are important for biology, then the cells should feel the forces through their adhesions to matrix and other cells, not at every point in the membrane. Just like if you put up a tent, like a pup tent, you put anchors in the ground, you put poles up, you put a flexible membrane, and then you winch it under tension and it's stabilized. That's tensegrity. But the forces go through the tent pegs. So to pursue this idea, I had to develop entirely new methods, and I reached out to mechanical engineers and chemists and other sort of physical scientists. And this culminated in a paper in Science in 1993. We developed what we call magnetic cell magnetometry. We took a magnetic bead here on the order of a few microns in diameter. We coat it with matrix molecules or adhesion ligands for different cell surface receptors. This is a cell viewed from the side. There's the nucleus. And that's a nucleolus, a little black dot in the center there. It's on a thread. And we apply high-level forces, nanonewtons of forces, and you can see that cells are very strong. They don't tear off when bound to receptors that mediate adhesion to matrix that are called integrins. And when we twist on those receptors in this case, we can show that the cells get stiffer and stiffer as you apply more stress. So you can't, this is a twisting stress, a torque. We couldn't turn them past about 30 degrees. But if we put the same bead with a receptor for a ligand for a metabolic receptor like a acetylated LDL, the cells were very, very floppy, and the same with growth factor receptors. We also showed in this paper that you could take tensegrity models of sticks and string, put a weight on them, and they beautifully mimicked this linear stiffening response. And so this is the first time that we actually could show that cells, in fact, behave as tensegrity structures, and we could disrupt that with cytoskeletal disruptors. We also went on and used this to show that pulling on integrins, but not other receptors, activates signal transduction and even gene transcription in a force-dependent manner, meaning that these matrix adhesion molecules, like integrins, function as mechanoreceptors. They sense force and transmit it over across the cell surface over a specific molecular pathway. Now, when you pull on cells with beads, you don't make them grow. So the question really is, does the forces that go to the cytoskeleton, if you have a high enough force, can you distort the cell, and will that control its function? And to get at this, I had an idea where I wanted to control cell shape or distortion independently of all other parameters that everyone in biology was focused on in terms of growth control. So I gave them saturating growth factors, soluble factors in a defined medium, no serum. I wanted them to adhere to high density of matrix, but I know matrix controlled cell shape because cells stick by binding and pulling themselves out against it. So what I wanted to do is to engineer little adhesive islands coated by a high density of matrix, separated by non-adhesive regions, like you could think of like Teflon. And so the idea is that if I had a big circle, a cell would spread and spread and spread and flatten until its adhesions were no longer resisted by the Teflon-like region, and it would be flat like a pancake. But if I made a smaller island, I have a cupcake. If I have a tiny island, I have a golf ball and a tee. And the only difference would be how far they stretch. And we accomplished this by collaborating with a famous chemist, George Whitesides at Harvard, who was developing an inexpensive way to make microchips for the computer industry. And the way microchips are normally made is you use photolithographic etching. You have a silicon wafer, you put a photoresist polymer, you expose a pattern with ultraviolet light that dissolves or polymerizes particular regions. Let's say it dissolves the circles, and then you can basically etch in circular patterns onto nanometer scale. What George did is the same thing, but when he made this pattern, he poured, and all that has to be done in a clean room in white bunny suits, very, very expensive. What he did is he poured a polymer of a liquid polymer called polydimethylsiloxane, PDMS, which is silicon rubber, in a liquid form, polymerized it, peeled it off, and amazingly, you retain surface resolution down to 60 to 90 nanometer resolution. You then can put either proteins, or originally we put self-assembling monolayers of alkane thiols on and stamp them onto gold-coated chips, or now we could do it onto any substrate. And you can literally create patterns from one stamp not in a clean room, again and again and again. So we did this by putting down extracellular matrix at high density to create those islands. And now, and this is where I got involved into the world of nanotechnology. In fact, it was probably one of the first major papers in the field of nanobiotechnology. When we put human endothelial cells on these islands, on a big island, they spread like a pancake. On a little island, they were a golf ball on a tee. And the reviewers said, you can't really say it's due to distortion because there's more total matrix contact area here. But I knew that cells are not glued to the matrix, they're spot welded through focal adhesion. So we made many small dots that the total matrix area is the same as this small circle, but the cells spread from dot to dot, almost like a suspension bridge, and stretch to cover an area similar to the cells on a big circle. And using this, we're able to show that as you increase cell distortion or spreading, cells go through the G1S transition and proliferate. Whereas if you round them and they're still attached, they go through a suicide program known as apoptosis and die. And we could even show that they were binding growth factor receptors, and the signals are generated across the membrane, but the cell interprets that to go grow or die depending on the shape. And these were published in 2 breakthrough papers in Science, and the 1997 paper has over 5,000 citations at this point. Now, we then made lines of matrix, because these are capillary cells, so they could have cell-cell contacts. And now you can see that they differentiate. They neither grow nor die, and they form a hollow tube with a lumen down the center. So we can switch between growth, differentiation, or apoptosis just by how far cells stretch. And again, no time to go into all we've done, but the point is that this has led to a field known as mechanobiology, and with the idea that mechanical forces are as important as chemical genes, and that they are in fact key regulators of normal development as well as cancerous tissue growth and development. So this was my world until I founded the Wyss Institute in January 2009, 12 years ago. And the mission of the Institute is to transform healthcare and the environment by emulating the way nature builds. Basically, we're trying to flip engineering from taking engineering principles, trying to solve biological or industrial problems, to leveraging biological principles to develop new engineering innovations. And our vision is that breakthrough discoveries can't change the world if they don't leave the lab. So we're not just a research institute, we're really a translation institute where we translate disruptive ideas that we develop into commercialized products so they have near-term impact. Now, when we started, we were given the largest philanthropic gift in Harvard's history at the time of $125 million from Hans-Jörg Wyss. We've now doubled and tripled that since. But when he gave it, he said that I don't want you to do incremental advances like NIH, NSF fund. I want you to take on incredibly high risk problems that could have a potentially huge impact. And the biggest problem that I could think of at the time is that the drug development model is broken. It takes over $3 billion to go from a discovery at the bench to take something to the clinic. And there are many reasons for why this takes so long and it's so expensive, but a big part of it is the failure of animal models to predict. These animal studies slow the process. They can take years. They're expensive. There are ethical issues in terms of animal lives being lost. But the real problem, as shown at the left, that's comparing the bioavailability in drugs measured in dogs, rodents, or primates versus what they found in humans. And it's like a random, buckshot graph. But the real problem is that the results often don't predict clinical responses, and they could be wrong over 70 to 90% of the time, depending on the area. So when we formed the Institute, I created one of the major platforms to focus on developing what we eventually called human organs on chips. These are engineered microchips containing living human cells that reconstitute organ-level functions, not cellular tissue, but organ-level functions to accelerate drug development, replace animal testing, and advance personalized medicine. And the first success was in Science in 2010. We called a human breathing lung on a chip. Now, we're not trying to engineer a whole organ. We're trying to build a major functional unit, and I like to describe them as a living three-dimensional cross-section through a major functional unit so that we have a window on molecular scale activity inside living cells, inside a tissue and organ level context. And we started, the functional unit was the alveolus or the air sac. Now, as most of you know, the alveolus is where you have gas exchange, aerosol-based drug delivery, pneumonia, metastasis, et cetera. It's a relatively simple structure in that if you go to high magnification, there's air, a single layer of lung lining cell, alveolar cell, a flexible extracellular matrix, the basement membrane, and then the capillary cell, and then there's blood. But what it doesn't show you is that this is incredibly mechanically active. And we know from respiratory physiologists that the mechanical breathing motions are absolutely critical for development and function. So if I were to try to distill this down to its most elemental design principles, what makes an organ? That's two or more different tissues that come together and new functions emerge, with one often being a vascular tissue. In this case, we believe that dynamic flow of air and blood would be important, and cyclic breathing motions. And so I'm now going to show you an animation of how this works. So the chip, as shown at the top right, is the size of a computer memory stick. It's optically clear, made out of silicon rubber. In the middle, there are 3 hollow channels, each less than 1 millimeter wide. The middle one is cut into top and bottom by a thin silicon rubber membrane with engineered pores. It's coated with extracellular matrix. To make the alveolar chip, we have lung epithelial cells, human lung capillary cells. We just created the alveolar capillary interface. But the trick is we have cyclic suction through these side chambers, and everything's flexible. So now we get stretching and relaxing at the same rate and degree as when we breathe. We then can introduce air over the top to create an air-liquid interface like in the alveolus. And we could have medium without immune cells, or even whole blood if we put capillary cells on all 4 sides through the bottom. Now, if this were to work, it should mimic organ level responses. So imagine you have an infection, like a pneumonia. The bacteria stimulate the lining cells to put out cytokines, chemicals that stimulate the capillary cells to now become adhesive for white blood cells, which normally just flowed by, but now they stick and roll and migrate across and then engulf. And that's homeostasis. And now I'm going to show you imaging in the device. So what you see next are the white blood cells labeled white, fluorescent. You can't see the endothelium, and then the epithelium, the lung lining cells are behind the screen. To begin with, they just flow by because it's healthy. And these are fresh white blood cells. We took them out of my postdoc, so we know that. And now if we have an bacteria on the other side, there is that signaling, and they're pulled out under flow, which is very important. That's what happens in vivo. The cool thing is you could do any imaging you could do in vitro or in vivo. So I'm going to show you high magnification. That's one white blood cell, a neutrophil. Between these 2 endothelial cells that are unlabeled, it goes underneath, finds the matrix-lined hole, wiggles its rear end out of focus to the other side. Now I'm going to show you by phase contrast it coming up out of the epithelial lining cells. And now I'm going to show you the white blood cells in red and the bacteria in green labeled with GFP. And you see them being engulfed. So you just watch the entire human inflammatory response in this little rubber chip, human. Now, I don't have time to go into this, so I will just tell you that using this one chip, we have modeled diseases such as pulmonary edema, pulmonary thrombosis. We've modeled drug toxicities, even when they've never been seen in animal models, but led to death in human clinical trials. We could mimic that in the lung chip. Drug efficacy models, we discovered new therapeutic targets. There's even a new drug in phase 2 clinical trials that in part was supported by results that we found in this assay. We've also integrated immune cells, and we've integrated connective tissue cells as well. So you could add, it's like synthetic biology at the cell, tissue, and organ level. You could start simple. If you mimic an in vivo response, it's simpler than you thought. If not, you have to add more complexity. As you heard at the beginning, we've won numerous awards. I am most proud of the International Design Award. We beat out the Google car and a Frank Gehry building that year. Also, the World Economic Forum named this as top 10 emerging technologies a few years ago. My wife is most excited about the Museum of Modern Art, where it's in the permanent collection, and I'm a permanent member now of MoMA. And because of that, I get a discount at the gift shop and the restaurants for the rest of our lives, which is a great thing. Now, we've gone on and we've built human lung airway chips that reconstitute mucociliary clearance. We've built intestine chips, and now we're using cells that are from patients coming from organoids that we break open and put on the chips. They spontaneously form intestinal villi. And we could do things that no one else could do. For example, in the intestine chip, for the first time, we can grow microbiome, living microbiome, in direct contact with human living intestinal cells with the same complexity that you see in the microbiome in stool or in vivo. And this is important because microbiome is a major paradigm shift in medicine. It's important for health and disease, but if you put it on a culture dish statically, you get contamination and they die. Here, we can get a dynamic equilibrium and we can study their contribution to both health and disease. But the ultimate goals of these chips is to recapitulate human pathophysiology and predict human responses to drugs using clinically relevant dose exposures. Often in vitro, you just bathe cells in a drug and it doesn't have a lot of meaning for patients. And we recently had a breakthrough here last year in January, paper on the human bone marrow. We take cells from bone marrow from a patient or from blood, we mix them in a gel with bone marrow derived stromal cells. We have endothelium on the other side, and you get beautiful blood formation, white and red blood cells over a period of months. AstraZeneca, the pharmaceutical company, came to us with a challenge. They had a drug in clinical trials that had very unusual toxicity profile. The same drug given over 2 hours, over 48, either had neutropenia over 2 days, white blood cell count low, or neutropenia and anemia. And they couldn't mimic this in animals. And so the first thing we did is they had measurements of changes in blood levels over time in patients shown in gray, the pharmacokinetics or the drug exposure profile that's not bathing statically, but varies over time. Because we have flow, we could precisely mimic this pharmacokinetic profile. When we do that, we precisely mimic the reduction in white blood cells and red blood cells at 2, but not 48. And this cannot be seen in conventional cultures. We could also mimic recovery, which is very hard in animals. I'm not showing it, but using the bone marrow chip, we've also taken cells from a patient who had a very rare genetic disorder of marrow called Shwachman-Diamond syndrome, and we could beautifully mimic that on chip. And also, we even got insight into mechanism that no one's seen before. This is important because it's very hard to do clinical trials when you only have a handful of patients in any one location, but we might be able to facilitate that with chips made with cells from patients all around the world. Lastly, we had 2 companies come to us, pharma companies, saying, will you make chips with cells from rats and dogs? And I thought, well, we want to get away from animals, but they pointed out that for the regulatory approval by FDA or in Europe as well, they have to do animal toxicity studies for liver toxicity in rats and dogs. And so we made chips. These chips have hepatocytes, primary liver sinusoidal endothelium, primary Kupffer cells, primary stellate cells in the right locations. And we found that human, dog, and rat do in fact differ and can predict the differences that are seen in the animal models, which may help us in the future go to human more quickly. And this is just the tip of the iceberg. Here we have over 15 different organ chips of every type of organ you can think of. But when I first presented this in a review in 2012, I said, because we have endothelium line chips with 2 channels, that we might be able to create a human body on chips. Imagine if you have an oral drug through the gut chip, watch it be absorbed, pass it by flow to the endothelium line lumen of the liver chip, watch it be metabolized, watch it peed out by the kidney chip. Do you have heart toxicity? What does it do to the bone marrow, or aerosol drug to the lung? We've now done this published in January last year. We use a robotic sampler to do this so that we have greater versatility and can take a sample out for mass spec analysis to get drug levels or cytokine analysis, and then another drop goes to the next chip. And we were able to, using an automated instrument, we can keep gut, liver, kidney, heart, lung, blood-brain barrier, brain, skin, bone marrow, and pancreas chips alive for a month and functional while passing liquid from chip to chip to chip. And this is a collaboration with Kit Parker, who did the heart and brain chips. And we did this with a common medium blood substitute. Now, the reason we were funded to do this was the idea of predicting human drug pharmacokinetics in vitro, and to have the ability to do in vitro to in vivo translation of these parameters, so you could, for example, save time by designing clinical trials beforehand, early. So to do this, we did a first pass model of a gut, liver, and kidney chips, which is where most of the clearance and processing occurs. And we integrated an arteriovenous reservoir to give mixing, so you could take samplings from it, so it would be analogous of taking a venous blood sample. And what you're really trying to do is you want to be able to predict key PK parameters like the maximum plasma drug concentration, or Cmax, and the half-life, T1 half, at which to get the maximum levels. And we have now done this as well. We linked these chips together. We did oral drug nicotine through the lumen of the gut chip. And we also did an intravenous drug, cisplatin. And here we used a bone marrow chip to look at pharmacodynamics, the toxicity in the marrow. Because it is not oral, we could take out the gut chip. And with this, at the right, you see the dotted lines are predictions from our measurements in vitro, which we combined with computational modeling done in collaboration with a company called CFDRC. And this is the prediction of what the changes in drug levels would look like in time in dotted lines. The data with data points and error bars come from 2 papers published 10 years ago with different doses of nicotine, either oral or chewing gum, or pouched tobacco, or cisplatin IV at 2 different infusions, and we precisely predict these clinical drug pharmacokinetic parameters. So to summarize, our 2-channel chips can faithfully recapitulate human pathophysiology, mimic human responses to drugs and radiation, which I didn't show, using clinically relevant exposures, quantitatively predict human drug PK parameters. And so we think that they're really now ready to be integrated into the drug development pipeline. And this is an example of a technology translation success at the Wyss Institute. This was a startup in 2014. We had CEO, CTO, CSO, all were our staff who moved out. 19 staff members moved out, and the company is now 100 staff selling products internationally. Now, before I end, I have some examples of other bio-inspired technologies that have come out. Another big challenge that to take on was like, what are the biggest killers of man? Well, heart attack, stroke, atherosclerosis, and they all share the fact that the cause of death is usually due to vascular occlusion, often due to blood clot of a major vessel. And now there are great clot busting drugs out there, such as TPA, tissue plasminogen activator. But the problem is that only 4 to 5% of patients are eligible, because you can also have bleeding into the brain when you give them. And they have to do it within 3 hours of the injury, and it has to be done in a hospital. So we thought, what if we can target that drug only to the clot site? And then we thought, like, how would we do that? And then we thought, well, how does nature get platelets to that site? And it's due to shear stress. Narrowing of the vessel increases fluid shear stress, a mechanical signal that activates platelets to become sticky at that site, and that's why you get a clot. So what we did is we built artificial platelets. So we took some PLGA polymeric nanoparticles, about 180 nanometers. We used spray drying to make a hollow sphere made of them, which is the size of a platelet. And you could think of these like a wet ball of sand. Wet ball of sand, you could shape it, it's round, but if you shear it in your hands, they fall to grains, and we could tune these so that they fall to grains only at abnormally high shear stress. So imagine you have a patient who has a stroke. In the ambulance, you can inject these into a peripheral vein. They travel around like platelets. They come to a narrowing of the vessel. The shear stress goes up. Now they deploy into small nanoparticles. There's less drag force. They have TPA that binds to fibrin. And the idea is that they would dissolve that clot. And even if a bit of it flew off, they would keep dissolving it like an onion skin. And incredibly, we showed this works. We had an animal model of pulmonary embolism. We inject a clot Then after injecting the clot, we inject the nanoparticles. And as you can see here, that 100% of the animals die without the injection in 45 minutes and 80% survive if we inject these nanoparticles coated with TPA. But what is not shown, or what is shown in yellow, is that we did this using 1/100th the dose of TPA. And it's almost a homeostatic dose, and pathic dose. And so this really raises the possibility there's no systemic side effects with this application. Another example is sepsis, one of the biggest killers worldwide. Over 50% of all hospital deaths are due to this. This is secondary to infection. What happens is it stimulates an inflammatory response that leads to multi-organ failure and death. Now, what stimulates that response are the release of things called pathogen-associated molecular patterns, PAMPs, that are the endotoxins and the surface inflammatory molecules on various pathogens. Now, we had an idea, leveraging off the work we were doing in organs on chips, to develop what we called the dialysis-like therapeutic device, where you could have blood go in one side, you could have sterile saline in the other. If you have laminar flow in a microfluidic device, you don't get mixing, there's no turbulence. So the idea is we could use magnetic beads coated with ligands for pathogens and pull the pathogens out and the PAMPs out and let the cleansed blood go back to the patient. And this is the device. Put the magnet on, the beads are fluorescent. You see the magnet pulls them across. The beads go to a discard and the cleansed blood goes back to the patient. The question, and I should say that we did this with beads coated with antibodies to E. coli, and it worked beautifully, and it was a cover of a journal. But I was trained in medicine as well as a PhD, and I know that you don't know what the infection is when a patient comes to the emergency room, so you don't know what antibody to use. So we started to think, could we use something more generic? And I knew from med school that in our body we have generic pathogen capture proteins called opsonins, and the most potent one is, the most generic one is called mannose binding lectin, MBL. It binds to complex carbohydrates called mannans on bacteria, fungi, viruses, parasites, and toxins. Normally, it mediates phagocytosis and activates complement, coagulation, inflammation. That would be no good. So Mike Super on my team, who came out of the biotech world, took this huge protein, genetically engineered a small version that has the carbohydrate recognition domain and then an FC region from the IgG antibody domain, because that's used to make therapeutic antibodies. It gives you a much longer half-life. It's very easy to purify. And this spontaneously forms a larger complex. Long story short on this one, it binds to over 100 different pathogens and toxins of every class: fungi, gram-negative and positive bacteria, viruses, including, we just learned this year, SARS-CoV-2, various parasites, mycoplasma, toxins, etc. So we, at the Wyss Institute, we made a little rat ICU. We have tubes going in and out of the major vessels. We put the beads in coated with FCMBL. We magnetically purify the blood, and the blood goes back to the rat patient. And we found that if we inject endotoxin into these rats, all of them die in about 5 hours. And if we put them on this device, 85% survive. And we actually prevent the inflammatory cascade. Now, more recently, we simplified this to try to go towards patients. We just take a conventional kidney dialysis cartridge, coat it with FCMBL. It works just as well. And we are about to enter human clinical trials for COVID-19 patients in the next month or so. We also found that because we could capture pathogens, we realized that we might have a rapid diagnostic. So we just put those beads into blood out of a patient. Then we use a sandwich assay with another MBL with horseradish peroxidase, and it tells you whether there are PAMPs in blood. Now, the PAMPs could be, you could have live bugs, which is the way most people look for blood culture, of which case they often find 80% are blood culture negative, even though they have sepsis. And that's because the body is trying to kill the bugs with immune response, or you're taking antibiotics and they're killing the bugs. But the PAMPs that are released when the bugs die is what stimulates the sepsis inflammatory cascade. We measure PAMPs. So we did this, I'll focus on humans. We did 150 patients, and we found by measuring PAMPs, we could have 85% sensitivity and 89% specificity of diagnosing sepsis within 1 hour of a patient walking into the emergency room at Beth Israel Hospital here in Boston, whereas the blood culture only had 18% sensitivity. Like I said, 80%, it could not detect that there were no live bugs in these patients. That is also going to clinical trials within the next month. Now, another last example comes from the non-medical world. At the Wyss Institute, we formed thinking that we'll learn from non-medical and affect medical and vice versa. Joanne Eisenberg was inspired by the pitcher plant in Africa. I'll go back. She was trying to develop ways to make airplane wings non-sticky so that ice wouldn't stick to it. And she was inspired by the pitcher plant in Africa, where insects crawl all over it when it's dry. But when it gets wet, there's a very thin layer of fluid that makes it incredibly slippery, and they literally slip into the center and it eats the ants. She realized that that was due to the nanotopography of the surface. She then made nanotechnology to make artificial surfaces with similar nanotopography. And as you're going to see here, you get a very humiliated ant in that it also cannot stick to this surface. She then took crude oil, which is one of the stickiest materials in the world, as you probably know from where you are in the world. And not only does it not stick to these surfaces because it's liquid, but you can injure it like with a razor blade and it self-heals because the fluid just fills that spot. And this won the R&D award in 2012, and now that's now being commercialized for company Advanced Surface Technologies, where, for example, it's being painted on the hulls of ships to decrease drag force and increase energy efficiency. We took this and tried to make surfaces that would prevent blood coagulation for medical devices. And if you take a normal medical grade material, you put human blood without coagulant, it just clots right away. You coat the surface with our version of this, and it just beads right off. We then went to the hospital. We took an arterial venous shunt that's used clinically. We coated it with this coating, and we could put it in pigs for 8 hours without any anticoagulant, no heparin, and it does not clot. And that was the cover of Surface Nanobiotechnology. That's being commercialized by a company called FreeFlow. And to end, this is really just the tip of the iceberg. You know, I'm showing you some of my examples, but the Wyss Institute has 3,000 patents. We're responsible for 25% of the intellectual property of all of Harvard University and 25% of the startups, with now only 11 core faculty and 15 part-time associate faculty. It's really quite an amazing place. I do, however, as Harvard faculty, have to disclose that I am a board member, chair of the scientific advisory board, or hold equity in the 3 companies I mentioned. So don't believe anything I said, although I can say that most of that data was generated before we formed the companies. To end, design. Design is a nebulous term, but really it's about creating solutions, no matter what field you're in, and often doing more with less, being incredibly efficient. And there's no doubt that the organ chip is a design. It not only won the design award, we have scores of patents on it, and that's a patent drawing there. And it really involves not only design, but doing more with less. And as you heard, it's won multiple design awards. And in addition to MoMA, it's been in many different collections, including in your part of the world, as shown here at the King Abdulaziz Center for World Culture. And so science is even having an effect on the design and art world. And even another example is that even my tensegrity structures have been exhibited in museums. This is the Martin Gropius-Bau Design Museum in Berlin, where that's my cell model with the nucleus, and my first tensegrity is right there behind glass. Now, the reason for all this, and what has inspired me from the beginning, and I'll never forget this quote from Buckminster Fuller. He said that nature has no separate departments of biology, physics, chemistry, or art, yet our schools teach them separate. And it's really a horrible thing that we do to our young people, because this is a truth. And I think to really answer questions that are tough, we have to bring full force to bear on each problem. And I hope, if there's any message, it is to find your own path, but to not be limited by artificial boundaries that are put up by people around you. And with that, I will end. I invite you to our website at the Wyss Institute. I think you will enjoy it. And thank you very much for this opportunity.
Thank you very much, Professor Donald Ingber, for this is very exciting talk. We'll take a few moments for Joe to put up his sketch and show it to us. Joe, would you like to put your sketch up?
It's a hard one, I would think. I gave him a lot. Cool.
Oh, that's really cool.
That was fast.
That was really fast, true, actually.
No. I'd like a copy of
And-
that at the end, please. Thank you.
Thank you very much. It's capturing science too very well. We'll open the floor for question. We've received quite a bit of questions. Maybe I will take the liberty of the first question. How do you link the mechanical forces and shape change to chemical signals? Are we talking about distribution of molecules in the surface, actin filament redistribution? How these mechanical properties are translated to signaling?
So at many different ways, at the molecular level, as I said in the beginning, I learned in molecular biophysics, the shape of the molecule determines its function. And if you deform a molecule with optical tweezers, you change its enzymatic activity, or you change its motor activity, or if you unfold a protein like fibronectin, you expose binding domains that cause fibrillogenesis. And that's why when cells pull on matrix, you get fibrils along tension field lines. Inside the cell, when you pull on integrins, you deform the internal domain of the integrin, which is literally transmembrane signal transduction, is causing a conformational change in a receptor across the membrane. It can do that by having a chemical bind it, and that causes internal restructuring, or it could be done by pulling on it, and that causes mechanical restructuring. Or it could be electrical, like in a nerve, and it could be electromechanical restructuring, but it always is mechanical. We've shown that when you pull on integrins, within 5 milliseconds, you activate an ion channel that lets calcium in. Calcium stimulates a whole set of cascades. We've shown that you activate G proteins, small and large, in the focal adhesion, where the forces are concentrated. We've shown that the growth factor receptors are in the focal adhesion, and that, for example, inositol-lipid signaling, there's a synthesis and a degradation, and one's controlled by the binding of the integrin and one by the growth factor receptor. We've shown that you cause, cytoskeletal polymerization and reorganization. We've shown forces go to the nucleus and deform the nucleus. That people have shown that causes calcium influx to the nucleus. Other people who've shown that you unfold the chromatin, and that changes exposure to regulatory factors in the nucleus. So the point is that the cell actually makes its decision at the whole cell level, because I can turn on local signals and it's round in suspension and it doesn't grow. So that same signals, when the cytoskeleton changes and the nucleus changes, now go into proliferation. So it really is forces acting to restructure signaling scaffolds, complexes, nuclei, but doing it, nature evolved so the forces allow you to do it in a coordinated way. Just like, if you have two clocks ticking at different rates in a room, the force over the floor makes harmonic coupling. You get that mechanical, the cell's not tearing apart. Everything is done in a coordinated manner. That's what tensegrity does. If you have one tensegrity here and one tensegrity here, and you link them by tension, they're now one bigger tensegrity, and everything reorients. So we do this when I stretch my arm, the fascia stretches, the matrix, the cells, the cytoskeleton, the proteins that are on the cytoskeleton feel it, the ones in solution don't feel it, and everything is done in a coordinated way. And everything's remodeled to minimize stress. So if you pull on the cells, they turn into the force, they pull on the matrix, the matrix gets bundled, so it's stronger in that force. and that's how nature evolved to live in our physical world, basically.
Right. So we'll take more questions from the audience. The first question is, did you think about modeling how coronavirus affects the lungs?
Yeah, so I have a major grant from DARPA in the United States and from NIH. We've been working on COVID-19 since January of last year. I had been funded to use our lung airway chip to work on therapeutics for future pandemic viruses starting 3 years ago, but we were focused on influenza. When this happened, we quickly pivoted and we developed pseudotype virus with a CoV-2 spike protein, developed collaborations with people that have BSL-3 labs. Long story short, we have papers in review now that we have drugs that we've discovered on chips. Some of them are repurposed. They're already approved for other reasons that work all the way up to hamster models of COVID-19. And we found on chips, and other ones that are novel compounds that also work in vitro in human chips and in animals already.
Very nice, thank you. Another question is, when we study a certain drug using this approach, don't you take into consideration that in a human body, the organ is not working separately? I mean, in relationship to this, even when you construct the organ in a chip, you take, for example, some type of cells from the bone marrow, but there are other type of cells. So how is it a true mimic of an organ if you make a selection as well on the top of the cells that are taken from the tissue?
So that's a good question. I mean, these are not models, as I said in the beginning, of a whole organ. They're models of a functional unit. And then they're models that have to be fit for purpose for what you're modeling. So my lung chip, if I'm modeling pulmonary edema, the alveolus chip works well. If I want to model pulmonary fibrosis, I don't have fibroblasts or connective tissue cells. I can't model that. I have to develop a different model. So you have to validate the model specifically for what you're modeling. And so if I was trying to model response of the white blood cells to cancer drug toxicity and radiation toxicity, our model works better than any animal model and looks just like human. If I'm trying to model effect on lymphocyte development, I have to modify that model. So you really have to design the model and validate it. Again, we validate against human data and as much as exists. And so that is how you do it. Now, scientists often think they know more than they really know. They think that the review articles and the books are right? So one example is when I went to med school, I was taught if a patient has anesthesia and you have to get them on solid foods as quickly as possible because you don't want them to get ileus, which is overgrowth by bacteria that leads to sepsis and you die because the intestines stopped moving. And if you read the books and the textbooks, it says that's because fluid flow has stopped and there's overgrowth. So we took our intestine chips and we could stop fluid flow, or we could stop peristaltic motions, or we could stop both. And it turns out the fluid flow is not relevant. It's the mechanical motions that regulates bacterial overgrowth. So the point is that there are assumptions that are wrong, that people think you need complexity. We did a pulmonary edema and a reviewer said this should not be published because there are no immune cells with interleukin-2. Another reviewer said, this is amazing. They just showed you don't need immune cells for pulmonary edema. So the point is we can get insight better into mechanism of what's going on and design the models better because we can independently control all these parameters, which cells, which forces, which chemical gradients, oxygen gradients. But the questioner is right in that one model will not model the whole function. And that's why we are linking chips sometimes. You also could, artificially vary hormone levels, which we know are important, using microfluidics if you don't have the ovary, for example, or you can, or you can link it to a ovary on a chip if necessary. But it's not perfect, but you're comparing to animal models that are sometimes 96% wrong. So it's really the lesser of two evils here. Even the FDA told us that if you're just as good as an animal model, no better, but as human, and you're as robust, we will accept that in place of an animal model. So that you have to put it all in context.
Yeah, I mean, it will be a big thing if actually it does replace animal model that will drive science forward dramatically.
But it would be one model at a time because it has to be designed.
One more time. Yeah, yeah. I have one more question, then we'll go for a bit more broader question. The question is, what would be the main challenge of the organ on a chip technology to be applied for healthcare in the future and diagnostic? So how do you make it popular? How people can access it? Is it easy actually to access this as well?
Well, first of all, we have to commercialize this so that you can do this anywhere in the world and get the same results. So we have automated, the company Emulate has automated instruments and sort of plug and play, but still needs to get better and better in terms of automating more and more. The second thing is that this is not a 96 well plate. This still is, it's more like animal studies. You got to grow up the cells, you got to pre-differentiate them and so forth. I will say that, people are starting to talk about using it for personalized medicine where, your lung on a chip, or let's say your liver and intestine on a chip. If you have inflammatory bowel disease, you could test drugs for efficacy and toxicity at the same time and optimize it. I really think the big game changer will be in how clinical trials are done, because right now, they do thousands of patients, usually fail, then do statistical number crunching to find a subgroup that maybe responded better, and then they'll do a targeted trial and hopefully get approved for a narrow application. With these chips and iPS cells and organoids, you can imagine like taking a small population, narrow ethnic group, narrow genetic group, developing drugs for 50 people, and then using those 50 people in a clinical trial. That I think is a real game changer.
Yes.
And then we are working on identifying diagnostics biomarkers using these chips also. So that is also relevant.
Perfect. We'll take some broader question. A question is coming that, what is your suggestion for science students to gain inspiration from art?
Trust your gut feeling and follow what excites you. And if you ever think of that there might be a connection, pursue it, read about it, research it. If you ever have an opportunity to take on something that's outside of what you might not normally do, but potentially could be interesting, but you're thinking, oh, I don't want to, take on more, just do it. You can always stop. I think that's, I pursue every option for the most part. I'm overextended. And when I discovered that fungus that caused cell rounding, and this was shown in a Nova TV special, reenacted, there was another postdoc with me, and I was so overworked and overcommitted. I had 20 projects, and I showed this to her. She sat on the microscope and looked at it, and I said, I think this has probably got an angiogenesis. What do you think I should do? And she said, throw it out. And I was about to throw it out, and I said, well, and I put it in a cold room, and then I told Judith Folkman about it, and then we both grew it up. But, there's the throw it out mentality, or the like, well, I might as well keep the option open. And I think you got to do that. And specifically in terms of art, take art classes if you enjoy them, go to museums if you enjoy them. But it's art to me is more the world around you, and art, there's always an artistic expression of anything that you do in the world. So you can always look for getting different perspectives on the same thing you're working on. That's what I would say. Look, like I went to the art library, the physics library, and the chemistry library, and the math library, because I was at Yale for 11 years. I knew them all when I wanted to research tensegrity. And each one of them, I would see things that resonated. I can't hear you all of a sudden.
Sorry, I'm mutated. Another question on this line, what type of background studies and expertise you need to work in the field? Engineering, biology, arts? Do all your studies follow your approach of linking fields? I guess the question is more into the interdisciplinary approaches and this type of bio-inspired research.
So in America, I don't know how it is where you are, but in America, we have a liberal arts education, and that as an undergraduate, we take, even if you're a molecular biophysics and biochemistry major, which I was, and I was even doing a bachelor's and a master's simultaneously, I could take as much, art and literature and history and music and social science courses and languages, and then we decide what to specialize in, and then we get narrow. Now, what I have found in my experience, like I basically stayed an undergraduate and have done that my whole life. I find that if I have people that come to my lab as as a grad student or as a postdoc or as a visiting professor, that they're narrower and narrower and narrower. So the key is keeping, the interest you had as a little kid in doing everything when you went to school, staying in kindergarten the rest of your life. I always thought of Judah Folkman that way, is that I joined Judah Folkman's lab. He was a famous surgeon, famous scientist. The first thing he did is he brought a science movie out and showed me called Surface Tension. And we watched it together. He was a little kid. And it's basically don't feel constrained to please others and play by the rules. Yet you have to learn how to learn how to play by the rules, show you can play by the rules, and you could do it as well as anybody, but then you have a different perspective that you could bring to the problem. And if you do that and you can explain things that other people can't, then you have impact. If you just sit there screaming, saying, I know better than you, nobody will listen to you. And it's hard, even if you do have impact, believe me, because people are very, very antagonistic about tensegrity and mechanics for many, many years.
It's a really new concept when you think about it. And
it's new to you, but I've been doing it since 1976. So yeah,
exactly, or ahead of time. Yeah, it's a great way of communication with the surrounding. It's phenomenal. I will take probably one more last question, if you don't mind. What is the next big question or challenge you think as a blue sky research?
What is the next big challenge, you're saying?
Yes, and question.
There's more concrete things, like we're now leveraging all of our capabilities to really do drug discovery and development. But on a bigger global, the environment and sustainability and leveraging biological approaches and synthetic biology and, biologically inspired engineering to deal with climate change and the environment, I think is obviously the big challenge.
Great. I think we can conclude our session. We've taken quite a bit of your time. Thank you very much, Professor Ingber, for your time and this really inspiring talk. We've pretty much enjoyed it, and we've learned a lot. And I'm sure I have a lot of the other questions that came from, I think, from students. They were quite inspired about your way of thinking across different fields and learning from nature, integrating nature, build imagination, think big. So it was really, it left a very positive impact on many people, as I've received many comments from them. So thank you very much. We appreciate your time. Thank you.
That makes it all worth it.
Thank you. So I think we are by ourselves, or we've... I'm
going to go off. Thank you so much, guys.